All right, AP Bio students, let's talk about some genetics and some inheritance. This section is called Mendelian and non-Mendelian, and I'll explain kind of what the difference is. Mendelian genetics is more of the classical like dominant versus recessive traits. Not all traits are Mendelian. We'll kind of go into that in some math, like in statistics that we can do to determine whether we think a trait is Mendelian or not, or whether there's something else kind of going on. I mentioned this in the last video at the very end that when Gregor Mendel was doing his experiments with the pea plants, he didn't know about genes. He didn't know about DNA. That wasn't a real thing at that point. Or chromosomes or all of that. And that adds a whole nother dimension. But we can start with like his basic idea and then go from there. So let's go into all these terms. We went over a lot of these in class, so I'll be kind of brief. But yeah, a gene and an allele are alternate versions. So a gene is a specific piece of genetic information. Let me actually pull out, I want to pull out that work that we did in class real quick, just I actually think it's slightly more useful than this. I drew all over it, but you get the general gist. So yeah, we kind of went over all this in class, but I'll just run through it again with you. So a gene, for example, D is a gene. D is like just a random example of a gene. It's a piece of hereditary information encoded in DNA or RNA. In eukaryotes, it's definitely DNA. Some prokaryotes like, well, not even bacteria. Viruses have RNA instead of DNA sometimes. It depends on if they're an RNA virus or a DNA virus. So there you go. I think coronavirus, for example, I believe it's an RNA virus, but we'll get into that eventually. Anyway, it's a gene that encodes for information. An allele is different versions of that gene. So this is if this gene, whatever this gene controls, whatever trait this is associated with, whatever type of gene it is, these are two different versions of the same gene. This is the dominant one. Usually we use capital to represent dominant, and this one is recessive of that same gene. So those are the alleles. In terms of, well, that's what dihybrid means. Yeah, that makes sense. Okay. Anyway, so genotype and phenotype, your genotype, heterozygous and homozygous is good. So this is when you have the two different chromosomes they have the alleles for the different genes. If it's heterozygous, it means that they have two different versions, so the dominant and the recessive usually. If it's homozygous, it means that they both genes have the same allele. So it could be homozygous dominant or homozygous recessive. That's what we're talking about there. Monohybrids are organisms that are heterozygous for a specific trait, and dihybrids are organisms that are heterozygous for two different traits, for two different genes. So that's kind of where a dihybrid cross comes from. Yeah, two organisms that are dihybrid or heterogeneous for genes of interest. This kind of comes in when we get into Punnett squares. So dihybrids are kind of a thing. So for example, there is a great example down here of a dihybrid cross. We get these two organisms are heterozygotes for two different traits for both the shape and color of pea seeds. So this could be like a dihybrid cross. I think sometimes this can also be thought of as a dihybrid cross where you have the dominant, the heterozygous from both. Anyway. Genotype is the genetic makeup. So I would say a genotype, for example, a genotype here would be dominant A or, AA, capital A, or lowercase a. In the example from class, like this is a genotype, DD, or, that. Those are the genotypes. So it's like the actual genetic content, like what genes are present. Whereas the phenotype is what's actually expressed. So we figured out for these, if this is a dominant trait, all three of these genotypes are going to produce the same phenotype. This particular, the homozygous recessive will produce a different phenotype. So there's actually 3 different genotypes, but only 2 phenotypes. So that can make a little bit of a difference there. Recessive versus dominant alleles. There you go. Autosomal chromosomes are the non-sex chromosomes. So in human beings, we have 22 autosomal chromosomes, and we have or 22 types of of autosomal chromosomes, a total of 44 total. 22 from mom, 22 from dad. We have one set or two sex chromosomes, which are the ones that actually determine sex, which is X and Y in humans. In other organisms, like some fish and amphibians and like lizards and birds, it can be ZW. There's a couple of different systems for determining male, female. It's especially in organisms that are capable of the sex is like a biological sex is it's like a phenotype essentially. It's kind of more complicated than that because honestly there's a lot of stuff that goes into it, but there you go. For some organisms, the external temperature, external like factors can alter sex, and that's usually because of that's a whole other thing we'll get into. Wow. Anyways, that's notes 3, so that's coming. It's complicated is basically the issue. It's way more complicated than you think. It's not just X and Y. There's a lot more going on there. Anyway, so yeah, so there's that. So Mendel has 3 laws that are, he's got kind of 3 laws of inheritance, which are genetic segregation, independent assortment, and probability. So genetic segregation is that genes separate, so each gamete gets one allele of a gene. So that's the idea of having 2 copies that separate. So that kind of goes back into meiosis, essentially, and the idea of having 2 sets of chromosomes. The next one is independent assortment, is that the alleles are not always connected to each other. So in his example, like with here, the idea is that the shape and color of the seeds is not connected to each other. In Mendelian genetics, that is true. In actuality, that's not always the case. Sometimes different genes are connected with each other, or they're more likely to pair off in particular ways because of chromosomal inheritance, because these are not individual traits floating around separately. They are on a physical chromosome and they're near each other on that chromosome. So this is where we start getting into potentials for non-Mendelian traits. And these are, again, traits that don't work according to these laws. We can generally assume that most traits will work this way. And then we can actually do statistics. It's going to be called the chi-squared test, which we'll learn about. I don't think I want to go over that in this set of notes, but maybe the next set of notes we'll go over it, and we will definitely practice it in class a bunch. It's a statistical test to help you determine whether or not whether or not something is like going by Mendelian assortment laws. Yeah, so the final law is the law of probability. And this one is just the likelihood of inheriting traits. They fit certain ratios that are kind of based off of Punnett squares. So that's like this thing right here. So the chance of a dominant phenotype is 3/4. It's this idea that you can kind of add these percent chances together. So 75, in terms of a genotypic ratio, we have 25% of offspring in this scenario should be dominant, heterozygous dominant. 50% are likely, based on this, to be homozygous dominant, heterozygous, and then the other 25% are likely to be homozygous recessive. However, that's genotype. Phenotype, 75% are likely to have the dominant trait and only 25% the recessive trait. So yeah, that's the law of probability, essentially. It's the kind of the probability and chances of offspring having different traits. That's what the Punnett squares will kind of help us to figure out. So there you go, there's Punnett squares. It's just a way of organizing data and organizing all these different traits. So like, for example, this is a more complicated Punnett square because it's dealing with 2 different traits that are independently assorting. So there's a couple of different options. So yeah, so we've got these have 2 traits. So the dominant and recessive version of the shape and the seed color. And so there are 4 possible combinations. Both traits being dominant, the shape being dominant, but the color being recessive. The shape recessive, the color dominant, or both shape and color being the recessive version of the gene. And you can create this chart, which then gives you an idea of all the possible combinations. And you can go through and figure out all the percentages, which I think are already done down there. The genotypes and the phenotypes, giving you an idea of, what the probability of finding different seeds are. So what we're going to end up doing later, so this is the kind of phenotypic ratio. I called it like a frequency, but it's the same kind of idea. And there's also a genotypic ratio. And again, that comes from the Punnett squares. So it's pretty simple. It's kind of statistics, but like really, really, really basic statistics. So there you go. Coolio. This is a little explanation of how Mendel kind of set up this P experiment. So it's a pretty cool one. Okay, let's keep going. Awesome. Pedigrees are something we're going to go over. Pedigrees are ways of keeping track. It's not for genotypes, it's for phenotypes, of keeping track of phenotypes across families. So this can be for human beings, or it can be for any sort of creature or any sort of organism. That will kind of help you keep track of these things, and you can go through and try to figure out, based on these phenotype or based on these pedigrees, what type of trait this likely is. You don't always have an absolute answer from this. So for example, these ones have autosomal dominant or recessive, or X-linked dominant or recessive. Pedigrees can help you figure out if the trait in question is related to that. You kind of have to kind of piece through it, like go through and figure out how that works. In all 4 of these examples, the shaded in boxes are traits that are, it's it's like a phenotypical trait. So if this trait is present. So this one kind of makes sense if both parents exhibit a particular trait, it's probably dominant. That would make sense. And then these ratios make sense that of the 2 children, one would have the trait, but there's a 25% chance that the other child wouldn't if this is dominant. So this seems reasonable. If this were a recessive trait, then both children, both children would have to also have that recessive trait. It would be much more statistically likely. Same thing here with the recessive. If it's a recessive trait, then neither parent would have it if they're like heterozygous or have homozygous dominant alleles. So it is possible for one of their children for this trait to pop up if they both got they would actually both have to be heterozygous in order for that to happen. If it's X-linked, it's a sex-linked thing, so then it would affect males and females in different ways in that particular case. So yeah, recessive, it's more, if it's dominant, I think females are more likely to be affected. If it's no, is it the opposite? Dominant females. Yeah, I think so. If it's X-linked, because then the females will get it more often. If it's X recessive, then females usually don't display that trait, but males do. Anyway, it's kind of a little bit, you kind of have to like think through these, and we'll do some practice with them. But yeah, looking at these, it would be the type of question where I could give you a pedigree and you have to tell me what type of trait it is. If it's just a somatic trait, which just means a normal chromosomes, or if it's a sex cell related one. I think there kind of are Y traits as well. There's just not as many of them. There's a lot more X related traits because of the additional X chromosomes are just bigger and have more genes on them. Y chromosomes are smaller and have fewer genes. So it's less likely. But anyways, we're getting actually into types of inheritance, which we get back to. So we'll play around with these. You can't always prove anything with a pedigree, but you can there's evidence to support an idea. You can maybe eliminate possible options. So, okay. There's different kinds. Monohybrid is a typical kind of Mendelian trait. It's a trait that is controlled by a single gene. but there are also lots of polygenic traits, which are one trait that's controlled by multiple genes. Actually, it's further down here. There's an example of that further down with the multiple alleles traits with the color, the coat color of Labradors or Lab retrievers, which we'll get into in a second. That can be kind of cool. We've got complete dominance where there's one allele that completely dominates others, but there's also incomplete and co-dominance. Incomplete dominance is where the two traits kind of mix. So a great example of that is with these flowers, these snapdragons. You have red, white snapdragons. The red is dominant, the white is recessive, but a heterozygote actually is pink. So they both traits will show up and they kind of blend together. You also have codominance, and codominance, let me see, hold on. Skin color comes. Oh, multiple genes, sorry, also, this is an example and I didn't label it properly. There are 3 genes that contribute to human skin color. So that's a little chart that kind of goes through that whole thing. So that's what that is. So yeah, like melanin levels essentially. In skin, there's 3 different genes. So that's one of the reasons why we have such like a wide variety of skin colors, melanin pigmentation, because it's controlled by 3 different genes. So you kind of get a wide palette, which is also why certain families, like people have different, like relatively different skin colors, even like if you have the same parents compared to siblings, just because genetic variation can happen. So you can have a family where people have, yeah, just really different skin tones, different melanin levels. It's kind of interesting. That's cool. It's complete, got 3 genes that contribute to 1 trait. Although, you know, it's a lot of skin. It's like the biggest organ in our bodies, or epidermis. Anyway, back here. So incomplete dominance is, yeah, that one. Codominance is when all of the traits are expressed. So sickle cell anemia is an example of that, and so are red blood cells, or like, yeah, blood types, which we'll have a good example on the back. So the example here is that, oops, lights are out. 1 second. The example here shows the heterozygous, the homozygous dominant. Sickle cell anemia is a recessive trait. So the dominant, you would see no evidence of that. The heterozygotes, though, you actually see both. So you would see normal cells and sickle cells. And the recessive, homozygous recessive, you would only see sickle cells. So technically, like people, if you are someone who is a heterozygote for sickle cell anemia, is likely to be able to function pretty normally because enough of their blood cells are normal. They still might have some symptoms, but it's not going to be like very terrible. Folks with the recessive, the heterozygote or the homozygous recessive, it's a much bigger problem because their blood cells very few of their blood cells function properly. And the functionality is reduced considerably. So they have really big problems. Crazy. But anyway, that's kind of an example of codominance. Again, blood typing is also a great one, which we'll go into in a second. This is a cool example of multiple allele traits. So this is kind of neat. There are 2 different genes that are involved for dogs. There's the B trait and the E trait. And I don't remember which one's which, but I'm going to write them down. One of them is for coat color, so the color of the dog's fur or hair. And the other one is for pigment attachment. And here's what that means. So the coat color, there's 2 choices. It's either black or brown. Right? So you get black labs or chocolate labs. The pigment attachment is whether or not the pigment, that brown or black pigment, can actually stick to the fur. So if the pigment doesn't actually stick to the fur, which I believe, I'm pretty sure I'm correct, is the E, it's a recessive trait. So any dog here that has the EE for recessive, so that's recessive. and black is the dominant and brown is the recessive. So it's two pretty much complete dominance traits. If the dog has black labs are far more common than chocolate labs because the black is the dominant color over chocolate. So you have to have the chocolate labs. I'll put a little box. You can't really see here. I'll put a little box around the chocolate labs. This is a chocolate lab. This is a chocolate lab. This is a chocolate lab. Yeah, so it's only 3 out of what, 16? It's not a very high percentage of chocolate labs. They have to be homozygous recessive in order to be brown versus black. But they also can't be homozygous recessive for the E, which is the pigment attachment. So yellow labs don't have either of those pigments. The pigments produce, it just doesn't actually attach and stick to the hair or the fur. So they don't have any color at all. So then the actual the pigments doesn't really matter. The pigment the genotype for the pigment doesn't matter because it doesn't show up in the phenotype. It would for breeding. So for example, this yellow lab would be capable of producing a chocolate lab offspring, but these three, all 3 of these labs would be capable of producing a chocolate lab. This particular yellow lab would not. If you're into like breeding labs, then this would be something that would be interesting to you, I suppose. So you could figure out what type of labs you want, if you want black, chocolate, or yellow. Anyway, I thought that was kind of cool. Dogs. So that's a great example of just how these 2 different traits, it's also known as epistasis. 2 different traits can determine one phenotype, so 2 different genes. So yeah, kind of crazy. And I think the same thing kind of comes up with like human skin tones or colors. These 3 different genes kind of determine. I think it actually, I don't actually think it's like that. I think these 3 genes somehow between the 3 of them have to do. I think it's all melanin levels. I think it's all melanin, but I wouldn't. Don't quote me on that because I'm not 100% certain. So anyway, there you go. And then there could be more complicated things that come with that too. So who knows? Genetics, this is the big lesson I've learned from genetics, is that nothing is simple, and that there are traits that we can trace down to 1 gene, and it's so cool, or even a few genes. But like so many things about being, especially just being a human being and being like dealing with diseases or like mental traits, anything like with your brain, it's like so much more complicated. And this stuff is like just doesn't even apply because it's just so complicated. And there's so many things that like add up onto this. So anyway, it's cool, but not everything we can't like pin. I'm always very cautious about whether or not we can pin anything to do with humans on genetics, on like a specific genes. A lot of stuff we can, some stuff, no. Anyway, something to think about. Okay. Yeah, let's talk about a little bit about these things real quick. We'll get through these, and then I think we'll stop over here before probability. We might go into the fly eye example, or I might cover that in the next video. We'll see how I feel. Okay, so sex-linked traits are traits that are on, if they're genes on a sex chromosome. So this is a great example of an X-linked trait. So this is a gene that is on the X chromosome. So the Y chromosome, it's for hemophilia specifically, has nothing to do with this. So if the father is unaffected with the hemophilia, it looks like, oh yeah, hemophilia is actually a dominant trait. It's just, this is another thing just to kind of keep in the back of your mind. Dominant doesn't mean that it's always going to show up. It just depends on how often that particular allele is present in a population. So hemophilia is a dominant trait, but that doesn't mean that like everyone has hemophilia. The actual dominant gene is, this is a carrier mother. Oh, no, it's a recessive. I'm sorry. I got confuddled. I was gonna say, wait, that doesn't make any sense. It's a recessive trait. But there are some situations like there are some diseases that are dominant traits. They just don't, that particular allele doesn't appear in the population very often. So most people get recessive. So it can still be something that you don't see very often. That is not the case with hemophilia. It is in fact a recessive trait, of course, because it's a carrier mother and the daughters are unaffected. So if the father is unaffected, then all of the daughters are going to receive the X chromosome from the father. So meaning that they're guaranteed to not be affected. So you're going to end up with, even if the mother is a heterozygous carrier for that particular trait, it means that this daughter is not a carrier, so her children won't have hemophilia unless the father has hemophilia. But this daughter would be a carrier. So like 25%, half of 50% of daughters, so 25% of children, but half of the daughter, there's a 50% chance that a daughter would be a carrier. Someone whose offspring could end up having hemophilia. For the sons, if they all get the Y chromosome from the father, so it just depends on which one they get from the mother. So there's a 50% chance that a son will have hemophilia. And if the son has hemophilia and he has offspring, then it's possible that his daughters could have hemophilia depending on the mother. And if she has a recessive, homozygous recessive or heterozygous trait. If she's homozygous dominant, then they won't. So hopefully that kind of makes sense. Good times. But it's on the X chromosome. So this is something like we go back to those pedigrees to keep your eyes open for whether or not there seem to be only female offspring or only male offspring are affected by something. Then it probably is sex-linked. Y-linked is also really interesting. That's a little bit less likely, but Y-linked traits never affect women. Color blindness is, I think, no, color blindness isn't. It's an X trait. That's why men are more likely to be colorblind. So I don't actually know what trait this is, but this would be daughters can never be affected by Y traits because they don't ever have Y chromosomes. Yeah, mothers are never affected if it's on the Y chromosome. So what that would mean is that all sons, all male offspring of someone who's affected with a Y-linked trait will always have that particular trait. So, but daughters won't. And daughters won't even be carriers because it's all on the Y chromosome. One thing that's interesting here, and we'll have to look at this like through karyotypes or like microscopy pictures. Y chromosomes really are smaller. That's kind of why they're called Y. I think it has to do with the shape, sort of, kind of, but not really. But yeah, X chromosomes are a lot bigger and contain a lot more genetic information, whereas Y chromosomes are a lot smaller and contain less information. So generally speaking, like there's a whole bunch of genes on the X chromosome that doesn't even appear on the Y. And they do have some corresponding genes, but there's just, yeah, a whole bunch that don't even show up on the X, don't even show up on the Y chromosome. So I think Y-linked disorders or Y-linked genes are just like less common in general, and having issues with them I think is less common for that reason. Let me see if I put that in here. I want to check something real quick. Did I put in bar bodies? Did I forget to put bar bodies in here? Oh my God, I think I totally forgot. Okay, I'll have to add that somewhere near the back. There's this really cool thing, and I'll mention it in the next set of notes for sure. Females of all species, I think most anyway, definitely like mammals and things that have like animals that, or organisms that have XY systems. So females have 2 X chromosomes, and in every cell, one of the X's becomes inactivated because otherwise they have too much genetic information compared to males. And there's a couple other reasons too, but it just happens. So randomly, one of the 2 X chromosomes gets completely like just locked up, it just kind of shrinks and it just kind of compacts and never really unravels. It's why calico cats exist. It's kind of cool. And calico cats are only female. So, and tortoiseshell cats, I think also. It has to do with fur splotches. It's pretty dope. We'll have to include that. Let me add that to my notes. Let's see, where would I put that? I'll put bar body / it's called mosaic. Yeah, there's a cool video. I'll have to, but there it is down there. So I'll try to remember to include that. I always forget something in these videos or in these notes. But yeah, it's pretty cool. So calico cats. I won't forget about that. You can always ask me about these things and I'll tell you more. Okay, next thing is non-nuclear inheritance. We mentioned this earlier, but nuclear inheritance is chromosomes that are inside of the nucleus. However, there is DNA even in human beings, even in plants, even in eukaryotes that is not included in the nucleus. This is things like mitochondrial DNA and chloroplast DNA, because they are, it's part of the endosymbiosis theory, that those were once independent organisms that are now like sort of permanently symbiotic with eukaryotes. They were like prokaryotes at one point, independent prokaryotes. It's also the case for these things called plastids, which are just extra DNA that's usually in little circles like a plasmid. Plastids inside of, variegation is maternal, and I would put the word plastids here. Plastids in plants. I don't think eukaryote, like animal cells don't really have plastids as far as I know. But anyways, those are just kind of determined. They tend to be maternal. Sometimes they're not, but they tend to just be maternal traits that are only, that, you know, sperm cells have mitochondria, but they don't give them, that's not how mitochondria is inherited. It's only inherited by mothers, which is pretty cool because mitochondrial DNA, because of that, is more of a direct line. So you can trace certain types of ancestry better through mitochondria. It's a very expensive process. It's kind of difficult to do, but it is possible. I think they use I could be wrong, but I think they use mitochondrial DNA to determine that like some obscene percentage of people born in like what is now like Asia and Eurasia are all related to Genghis Khan. It's like something like 50% of all people in like that part of the world like are, you know, in the like, yeah, related to Genghis Khan, because yeah, during the Mongol horde ran around all of Asia and the steppes and Mongolia and all of that, and just did a lot of raping. It's great. Left behind a lot of offspring. It's kind of crazy the effect that one person can have. You should look it up. Genghis Khan. Everyone in Asia is related to Genghis Khan. Not everyone, but like a huge percentage. I think they use mitochondrial DNA to figure that out. I'll make a note of that for myself. Let me look that up. Hold on. I gotta find a piece of paper to write it on. It's pretty funny when I look up all these notes and it's just like weird stuff like Genghis Khan video. But I think it was through mitochondrial DNA that they figured that out. Anyway, more ramblings, just what you wanted. Makes these videos very long. It'll keep you busy. Okay, the other example is the variegated plants. I love this because I've got a lot of variegated plants, and I know that it's all based on the eggs from the seeds and all of that. So yeah, it's those plants that instead of being all green, have like some white on them. It's called variegation. It makes them look pretty. Those variegated plants also need more sunlight than non-variegated plants because they have white. That means that there's not as much chlorophyll. So they need more sunlight than their non-variegated relatives. I don't think you get too many white plants because straight up white plants don't have a lot of chlorophyll. It's kind of hard for them to survive, but I think it's possible. Sometimes they exist. But the variegated has both, and it is completely related to the egg cells. Has nothing to do with the sperm. Makes no difference. Okay, So that it's a good thing just to vaguely have in your mind. This is one of those things where it's like a little detail that like, yeah, if you forget it, it's not the end of the world. But if you can just vaguely remember, oh yeah, like some like non-nuclear DNA is probably maternal. It could be paternal. It could be from the sperm cells only, but that's not very likely. You never know. Okay. Let's talk about blood types for an example. Blood types are really cool. I might come back and do a little bit more at these because I'm kind of petering out right now. I just did the other notes and I'm tired. And I don't have that much to say. You're like, yeah, Mr. Drapkin, you just rambled on about Genghis Khan for 10 minutes. So clearly you've got a lot to say. Okay, so yeah, so the way that blood types work, blood types are actually has to do with carbohydrates. These are unit 1. Hold on, this is a unit 1 or a unit 2 thing. I think it's a unit 1. Carbohydrates on the outside of cells. I believe this would be this is a macromolecule for sure. This is like let me see what we come up with here. Lipids. Nope, it might be more of a unit 2 in terms of things around cells. It's not cellular energetics, that's for dang sure. Cellular structure and function. This would have to do with plasma membranes, embedded proteins. It's like a glycoprotein or a glycolipid. It is in fact a glycolipid. I even put that in your notes before, blood types. So these are glycolipids. They call them carbohydrates. Glycoprotein. Yep, that makes perfect sense. Glyco meaning carbohydrate, but also attached to lipids. That's like a transmembrane, like it's a tag essentially. This is a situation where there are actually 3 different alleles. Sometimes there's 2 alleles, sometimes there's more than that. In the case of like skin type or skin color, it was 3 different genes. This is actually 3 different alleles. So there's the A allele, the B allele, and then the none allele. The allele, like this is the complete recessive allele. So there's like dominant A, dominant B, and then nothing. And so depending on which alleles you get, which combination of which ones you get, it determines what your blood type is. So if you have the dominant A, you're going to be type A. If you have the dominant B, you could be type B, or you might also be type A if you have these 2 with the combination. If you have like A and recessive, it's just A type. Having O means only just having your homozygous recessive for like glycoproteins for the blood type carbohydrates. Yeah, so it kind of works out where like if your body has, if you are a blood type that has a certain glycoprotein, the A or the B, your immune system recognizes that. So if you have type AB blood, you can get a transfusion of A or B or O, because you have all of, like, your body is like your human immune system recognizes A and B as being part of your system. So AB blood types are considered the universal acceptors. O are the universal donors because anyone can take them. But O, if you have type O blood, you can only accept type O because your immune system says, hey, we don't have this A proteins or B proteins or carbohydrates. That's not a thing. Then that's kind of a problem. Your immune system will just be like, invaders, no good. We should talk about the human immune system, or just immunity in general. It's pretty cool. It's complicated, but cool. Anyway, I also want to throw this on here. There is also a RH factor, which is like another thing that's on blood that also helps determine this. It's like, but it's a separate gene. So this is one gene that determines your A, B, or O type, but then you can also be positive or negative for the RH factor. RH is just a simple two allele recessive dominant kind of trait. So you're Rh positive or Rh negative. If you have any Rh positive, it's going to be Rh positive. If it's homozygous or homozygous recessive, then you're Rh negative. And that does also affect like blood transfusions to some extent, I think. I'll be honest with you, I'm not an expert on like biology of like multi-cells. I like things that are one cell and smaller, and then if there's too many cells interacting with each other, I'm just like, what? Multicellular organisms? No, thank you. Bacteria, best friends. Anyway, I don't know much about Rh, but I think it's another factor that's determined. It's kind of cool. So your blood type is determined by two separate genes, one of which has two dominant and one recessive, has three different alleles. Most of these other genes we've been talking about before are all just like two alleles. So there's lots of options. I don't think there's a lot of examples of like four different alleles that give you completely different traits. That seems a little insane, but you never know. Fly eyeballs are another example of like, this is like a good example of like a sex determined trait. Maybe I will come up with something or we'll talk about it in class. I don't really want to right now, so I'm not going to. But yeah, it's a great experiment. It's Morgan's fly eye experiment. It's a classic experiment along with Mendel. So I'll find a video on it or something to throw up there for you. Next video, I'm also going to go over probability with you. This is, it's not as complicated as it looks. It's just like there's this crazy equation. It's the chi-squared equation. It's not that bad. Just knowing what it means is more vocabulary that goes with it. It's new, it's a little different, but you can handle it, no problem. Half of you are probably in pre-calc anyway, so this is nothing. And then we're just going to talk about some other little cool details like environmental effects on phenotype and this gene mapping thing and, you know, some other cool stuff. So it'll be a fun little set of notes that I'll record on a different day. So let's stop here. Oh, I want to show you videos. Never mind. We have to do that next. Again, my YouTube channel is not super organized for this particular unit. I tried, but I failed. In terms of all of this, there's some cool stuff here. There's Mendel's pea plants, very cool. This, where did genes come from? I have to watch it, but I think it was pretty cool. Oh, this is cool. What can DNA tests tell us about our ancestry? It's kind of neat. It kind of explains how like things get a little complicated and how if you and your sibling, if you have a sibling, get one of those like 23andMe tests, it might have different results because there's only so much these things can tell you. There's limitations of those tests, but they're cool, but they're also kind of limited. Gene mapping we'll talk about later. Let's throw that, we'll move that down a bit. This one could be great. I think this heredity one is very nice. This one's really cool, just specifically about Mendel, this lost and found history of science. Genetics is always good. Crossing over and mapping, always delightful. Alleles and genes, pedigrees, all of that stuff is good. Oh, here, a good explanation. Amoeba sisters, of course, amazing. Different forms of dominance and codominance and epistasis, that's dogs. And some cool Punnett square stuff, just in case, you know, that comes into play. And then we're also going to get into chromosomal genetics. I'm going to move this one up a little bit because Punnett squares. Very, very nice. And yeah, we're going to get into the chi-squared test a little bit later, but sex linked as well. All right, let's end it there, and I'll see you all for the next set of videos. Thanks for listening to my rambling.